ਸ਼ਾਂਤੀ ਸ਼ਾਂਤੀ ਦਿਓ ਮੈਨੂੰ, ਜ਼ਿੰਦਗੀ 'ਚ ਸ਼ਾਂਤੀ ਦੇ ਦਿਓ, ਬਹੁਤ ਹੈ। So, good evening, class. So, as promised, we will see us over the weekend on Saturday. I hope it's fine. And we finished our animal cell culture for today. I hope you don't have any lecture afterwards. And yeah, so that because we are going very, very slow, I didn't thought that it will take us so much time to come only 2 days work, and since we started. So yeah. So this is the stem cell culture works with the first stage of your animal cell culture is morula, and then which makes blastocyst, and from these blastocysts we have inner cell mass, which we grow in the embryonic stem cell culture, which could be divided into further cell lines as we are wanted. They could go for muscle cells, blood cells, neuron cells, intestinal cells, pancreatic islet cells, liver cells. They go into the things as they should be. As we provide the nutrients, the chemicals, the atmosphere that we provide, they will grow into the that cells. So stem cells, they have ability to replicate, self-replicate, and also differentiate to the multitude of different cell types needed within the body. So you have a self-renewal and differentiation. Two things could happen. In the self-renewal, you will have the pluripotent stem cells. On the other case, if they differentiated, they could go into three types of your cells, that is mesoderm, endoderm, and ectoderm. The mesoderms will produce RBCs, skeletal muscle cells, cardiac muscle cells. With endoderms produce your pancreatic cells, lung cells, and thyroid cells. Okay? And the ectoderm, they produce skin cells, neuron cells, and pigment cells. So in this way, we can differentiate according to their cell type. And again, this hematopoiesis. So you have from here your pluripotent stem cells. They could go for lymphoid stem cells to produce your B cells and T cells. Otherwise, they could self multiplication could happen. On other case, you have your myoblast producing basophilic erythroblast, orthochromatic erythroblast, reticulocytes, then RBCs. So this is the way of producing from your pluripotent cells to your RBCs. The same way from megakaryocytes, you can have your thrombocytes, that is your platelets. On other sides, eosinophilic, basophilic, monocytes. They are producing your monocytes and neutrophil, basophil and eosinophil. So all the your immunological cells, no? So all your erythrocytes, they all come into this section. And this is the normal cell cycle that we see. We having first this synthesis phase, DNA synthesis phase, then G2 phase where we check the cell size, DNA replication. Then there's a resting state for some time. Then comes your mitosis stage, as a spindle assembly checkpoint where chromosome attachment to spindles are checked. Then comes the G phase where they check the cell size, nutrients, growth factors, DNA damage. Then afterwards you have a resting state. So during this transition state, this is not required. Yeah, in the cell culture environment in vitro, so what are the basic materials that are required for the cell growth? So what are the basic needs? So far we have discussed yesterday all about the basic conditions that need to be maintained. But what are the now materials, one by one we will see, that are required in our cell culture environment. So first, the basic material that is needed for cell growth is substrate or liquid cell culture flask scaffold material, which chemically modified plastic or coated with ECM protein suspension culture. Then you need nutrients, culture media. We need environment like carbon dioxide, temperature of 37 degrees Celsius, humidity, and oxygen tension maintained atmospheric but can be varied. Then we need sterility, aseptic technique, antibiotics, and mycoplasma tested. So, what kind of media do we need? So, first of all, the basal media, the main media that we're using, it should have maintained pH of osmolarity and should have enough source of nutrients and energy source. So, what are the components? So, initially we need inorganic salts, which could maintain the osmolarity, membrane potential, some ions for cell attachment, and enzyme cofactors. So, for membrane potential, we need sodium, potassium, calcium, and we put phenol red into our medium to check the pH. So optimum cell growth pH is 7.4, which is a phenol red color. But if you see your medium color is changing to pink or yellow, if it is going yellowish, the yellowish means it's getting acidic. If it is going more pinkish, that is going more basic. So somewhere in the neutral is the color is red. Then the buffers that we use are bicarbonate and HEPAs. So bicarbonate buffered media requires carbon dioxide atmosphere, and HEPA strong chemical buffer range pH from 7.2 to 7.6. And this does not require carbon dioxide. So glucose energy source are needed. So cell culture environment in vitro. So components of basal media. Also, we need keto acids, that is oxaloacetates and pyruvates, which are intermediate in your glycolysis cycle. They maintain the maximum cell cytopolism, and they are added to media to provide an additional energy source. Then carbohydrates are required, vitamins, some trace elements as follows. Then some supplements are also required for cell culture environment, like L-glutamine, non-essential amino acids, growth factors and hormones, and antibiotics and antimitotics. L-glutamine, NERR, growth factors and hormones like insulin, antibiotics, and antimycotics. So they are quite essential. Then comes your FCS, FBS. This is also very important. So they contain mainly the growth factors, which helps to aid our cells to get attached to the surface of our test tube or the cell culture plates, and they neutralize the toxins, and they are being used from the very past time. So they are quite infectious agents. Variable compositions could happen, a bit expensive though. Is fetal bovine serum. It's written from Sigma company. And regulatory issues minimum risk. So first, but one thing you need to maintain with fetal bovine serum is you have to heat inactivate by temperature at 56 degrees Celsius for 30 minutes. Why? So that the complements and the immunoglobulins could be destroyed in them, and the destructions of some viruses like gamma irradiated serum. So overdoing it can damage growth factors, hormones, and vitamins, and affect cell growth. So how we do the cell culture in the laboratory? So all in all, in the past that we have seen here, so all these materials are very important. Your inorganic salts, your pH indicator, your buffer like HEPAs, which maintain the carbon dioxide atmosphere inside the cells, glucose, okay? So all these materials are already provided in this media. Everything is present in this media, in it. Earlier days, in our 40s and 30s and 40s, we need to maintain this culture. We need to make this by ourselves every time, fresh one. But now we have such good companies, such good system that everything is provided at once. Keto acids, carbohydrates, vitamins, trace elements, everything is there. The supplements is also available separately, so we can add them into our main media, maybe 1 microgram per ml or like in that concentration. As per our the cell culture required, the cell that we are growing, as per that we can change the things. So in order to start your cell culture, um, if our cells are frozen in a cell population, we need to isolate our tissues. Then maintaining the culture with aseptic techniques, right? And then subculture them, and then count the cells, and then you can put them again for cryopreservation. So in the cell culture laboratory, you get this typical cell culture flask in which you grow your cells. After done with the subculturing, passaging, you put them into minus 70 degrees Celsius. This is Mr. Frosty to freeze your cells. So what is passaging of cells? Basically in passaging, what we do, once if you're growing your cells in cell culture and they are overgrown and you want to have a fresh media, it's like in a pop, if we put some mouses in a container, they start breeding, and they will after breeding there will be so many mouses that it will be full of mouse after maybe 1 month or 2 months, so which will be not good for us. So what we do, we want to have a minimum population always, and we try to remove those mouses from that media and then keep the same level as it was before. That's the same with the cells. So we try to remove all the cells from the cell culture plate, and then we grow them again um, in a fresh media, in a fresh plate, and so that the overgrown things does not happen. So how we do it, we first check, like we have a confluency of 80%. So this is 70 to 80% confluence, what we are seeing here. And this is what happened at 100% confluence. If things are like this, so they will start to produce toxic material, and the cell starts to die eventually. And this is not healthy media, and it's become acidic. So you will see your media being colored yellow, and which is not good for your metabolic working space. So you need to wash in PBS to remove dead cells and serum. Trypsin digests protein surface interactions. EDT enhance trypsin activity. Then you resuspend in serum. Then transfer the dilute suspension to new flask, and the most cell lengths adhere in the 3 to 4 hours. So here the chart is there. You check how much cells you are confluent. If they're 80%, then good. Then you remove all the mediates present. Then you wash your cell confluency with PBS. Then you incubate with the trypsin. Then after incubating with the trypsin, you resuspend in the serum containing media. Then transfer to a culture flask. Yeah. So let's watch how this passaging cell is done. This will explain all the cell cultures actually, in a nutshell.
The growth of cells in culture follows a standard pattern. A lag after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate, or the cell density exceeds the capacity of the medium. Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration. Handle the cells with care during transport. For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution, such as DPPS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate, and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed. Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Use a north-south, east-west motion to evenly distribute the cells and transport the flasks to the incubator. When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You'll follow the same counting procedure for adherent cells using Trypan Blue and either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. Stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately, depending upon whether they are vented or not, and return them to the incubator. Every 3 weeks, gently spin the cells, remove the medium, and replace with completely new medium. This eliminates cell debris and metabolic waste.
So I hope this massaging is clear. Then after you're massaging the cells, you can go for cryopreserving your cells. Cryopreservation means you can put your cell for the long run into minus 197 minus 190 degrees Celsius for the long run. So your cells will be safe there. So you will have reduced the risk of microbial contaminations or contamination from other cell lines, and we the research that we conducting, it will be at consistent low passage. So you have a passage cells, you resuspend in the media, centrifuge them, then aspirate the supranatant, then resuspend them in the 10% DMSO, including your fetal calf serum. Then you transfer to the cryovial freeze at minus 1 degree Celsius, then transfer to your minus 90 degrees Celsius. So the cryopreservant that we are using is, which is precise mechanism unknown but prevents ice crystal formation. So this is the best thing to use, DMSO, which we freeze at minus 80 degrees Celsius, which is rapid yet slow freezing. So manual cell mount. So then how we count our cells after, for the passaging, for making our cell culture to count. So this is our hematocytometer looks like. Yeah, this is a specific hematocytometer which looks like, and you can count cells in the box, then repeat for other corners of the chamber. So we having these 1, 2, 3, 4 chambers. So we take one of the chamber in the corner and we calculate the average number of cells. Yeah, so we start counting your cells from each cell to all four. Yeah, and then we multiply with the dilution factor and then 1/10,000. How much is your sample is present. And there are also automated cell counts which do it automatically for you. So I will show you how a hematocytometer really works. HEPAS full form. Bache, HEPAS is very complicated term. It's something with the biochemistry. Hydroxyethyl, poro. I don't remember. It's not required to remember it, but I can Google search for you. Yeah, it's 4,2-hydroxyethyl piperazine ethane sulfonic acid, which is a zwitter-ionic sulfonic acid buffer agent, one of the 20 good buffers. Yeah. So hematocytometer.
A hemocytometer is a device that is used for counting cells. It's a modified microscope slide containing two identical wells or chambers into which a small volume of cell suspension is pipetted. We have already removed 100 microliters of our cell suspension and placed it in a microcentrifuge tube. Dilute the suspension by adding 100 microliters of trypan blue. Trypan blue is a dye that helps us distinguish between living and dead cells. The dye passes through the membranes of dead cells, so they will appear blue under a microscope. Living cells exclude and will appear mostly clear. Load both chambers by pipetting the suspension under the cover slip. Now place the hemocytometer under the microscope. Each chamber is divided into a grid pattern consisting of 9 large squares. Each square has the same dimensions and contains 10 to the negative fourth power milliliters of suspension. The rules for counting cells sometimes differ from lab to lab. In our lab, we count cells in the 4 large corner squares and the center square. Let's count the cells in the first square. 1, 2, 3, 4, 5, 6, 7, 8. So what about the cells that are touching the outside boundaries of the square? In our lab, we count the cells that touch the top and left boundaries, and we ignore the cells that touch the right and bottom boundaries. 9, 10. We need to count the number of both living and dead cells. Remember, the dead cells will appear blue. Occasionally, you will see artifacts, objects or debris that appear blurry and don't have a well-defined shape. This is an example of an artifact. We won't include it in our count. Proper storage, cleaning, and handling of the hemocytometer will minimize the number of artifacts. There are 10 viable cells and 1 non-viable cell in the first square. Now, the top right square. There are 9 viable cells and no non-viable cells. Next, let's count the bottom right square. There are 11 viable cells and no non-viable cells. And now the bottom left square. There are 10 viable cells and 2 non-viable cells. And finally, let's count the cells in the center square. Sometimes cells will appear as clumps or small groups. It may be difficult to determine exactly how many cells are in a group. The method of counting clumps of cells differs from lab to lab, so be sure to follow the procedure in your lab. We will count this clump as 2 cells. There are 14 viable cells and no non-viable cells in the center square. The total number of viable or living cells from all 5 squares is 54. The total number of non-viable cells is 3. Now that we have counted our cells, there are several calculations we need to perform. First, let's calculate the percentage of viable cells. Here's the formula. 54 viable cells divided by 57, the total number of cells, gives us 0.947. Multiply by 100, and the percentage of viable cells is 94.7%. Next, let's determine the average number of cells per square. We counted 54 viable cells. We divide 54 by 5 because we counted in 5 squares. The average number of cells per square is 10.8 cells. Now, let's calculate the dilution factor. The dilution equals the final volume divided by the volume of cells. Our final volume is 200 microliters because we started with 100 microliters of cells and added another 100 microliters of trypan blue. 200 divided by 100 is 2. Therefore, the dilution factor is 2. Next, we need to calculate the concentration of viable cells, the number of living cells per milliliter. Our average count per square is 10.8. The dilution factor is 2. 10.8 times 2 times 10 to the 4th power equals 216,000 cells per milliliter. We can write the concentration using scientific notation as 2.16 times 10 to the 5th power cells per milliliter. From our calculations, we now know the concentration of cells in our culture is 216,000 cells per milliliter, and approximately 94.7% are viable living cells.
Welcome back, friends.
Not Shomu. So I've shared this presentation also. Okay, that was automated. And this is the normal cell culture that happen in the bacterial and most of the cell line culture that we see. Initially we have a lag period, then it goes into this exponential phase, then they go for linear stable plateau. So this region we can say population doubling time, PDT. They are decreasing from like population is increasing totally double, and this is a saturation density, that this is the maximum population of our cells could grow. And these are on the x-axis we can see some days, that how many days it takes, and on the y-axis it's the cells per ml. So as I said, there could be some contamination could happen, cell culture contaminations which define the cell some elements in the culture. So one of the major contaminations are from the bacterial and yeast, viruses, mycoplasma, cross contamination by other cell culture, um, and another could be chemical contamination with the media, incubator, serum, and water. So, in order to control, first aseptic techniques, use of antibiotics, good housekeeping for everyone, nature, understand that how the contamination works, and after every month in the lab, we can have a monitoring program in order to check if there any cell culture is being anything is wrong with the cell culture. So, how human embryonic stem cells are derived? So in vitro, there is a day zero, and totipotent cells day three, and blastocyst cells day five. So traphoactum, this we have done already. So sorry for this slide. These slides we have seen already. Yeah, this one. These are the common cell lines that we use from day-to-day culture in cell culture. That is MCF cell line, HL60, HEC293, HeLa cells, primate cell lines, Vero cell line, COS7 cell line, and others such as CHO from hamsters, SF9, SF20 from insect cells. So we have MSF breast cancer cell line, HL60 leukemia, and HEX 293 human embryonic kidney, and HeLa as a Henrietta Lacks, and Firo and COS7. So we use them day to day, every time, quite often. So this is the organ culture. This is how it looks like. I think they're growing liver maybe. Here we having heart, kidneys, some organ. I'm not sure about this. This is question mark. This is somehow the kidneys feels like. This is the heart. So in this cylindrical tubes, we grow them. So under them, we provide all the required media that is required for them to grow, and it keep coming in and going out. So like this. So we maintain a natural conditions for them. So that's it from the cell culture point of view. How's that? Maybe it's because of the diluting factor they were using, but mostly we used to multiply with the dilute with the how much ml is in our sample, how much sample is diluted, and we multiply according to that. If it is present in 10 ml, then we multiply in microliter converting into 10,000. So in that way, we used to do it. So I think that's it from the animal cell culture point of view. That's all we have to do it. I want to make a short class, not a big class today. So we meet on Monday now. Tomorrow there will be no class, and from Monday onwards we have a fresh start about recombinant DNA technology. Yeah. So we will discuss about various molecular methods or RDA technology in detailed manner, very detailed manner. So then we go for recombinant DNA technology, RNA-based markers, DNA fingerprinting, restriction enzymes. So we cover all of them. The fifth day will be very interesting, where we will discuss about protein isolation and purification. So that day will be very interesting. Okay, thank you very much. Stay safe. Enjoy the weekend. Take care. Bye bye.